Okay, guys, I just wanted to take just a little bit of time and do a quick review before your test tomorrow. I'm going to start with water. Remember that water is a polar molecule, and it is referred to as ultimate solvent. That means that it will allow things to break down. It has a very weak hydrogen bond, which is between the oxygen and the hydrogen. And this is why it's so easily broken because the bond is kind of weak. One of the properties of water is called surface tension, and surface tension occurs because of the cohesion of the water. It sticks to each other and it allows it to spread out when something walks or tries to walk across it that has a less density of than water. Cohesion, water likes each other, so water molecules will stick to itself. Adhesion means that it will stick to other things. So this is why it sticks to surfaces of cars and um, utility poles outside, anything outside. Capillary action is when water's being pulled up from um, the root system of plants, and it will pull water from the soil, and it will travel up through the root system. A key component of this is the bigger the root system of a plant, the more water it will pull into the plant. And so plants that live in the desert will have a very small root system, and it's most of those plants store water in their vascular tissues up inside the plant itself. That's why a lot of cacti are short and round, and they have a lot of vascular tissue on the inside. Um, this property defies gravity. It means it moves in the opposite way of what it should or normally would. We're going to skip this part. We talked a little bit about um, solvents and solutes, and a solute is something that's going to be dissolved inside of a solvent, usually water. So an example of that would be lemonade mix, Kool-Aid mix, sugar, salt, and then our solvent, of course, is going to be water. Acids make hydrogen gas when they're mixed with water. An oxide, a base, makes an OH when it's added to water. Your pH scale is very important. Um, Water being neutral at 7, anything below 7 is considered to be an acid. Anything above 7 is considered to be a base. And then we go into our organic molecules. And our organic molecules, remember, are naturally made. They come from, that's their name, organic means naturally occurring. And then they're also called macromolecules. They're extremely large. The key component that all of these have in common is they all contain carbon. So we're talking about proteins, lipids, and carbohydrates. Remember the definition of a monomer. It's very small, usually 1 unit. And then polymers are all of these monomers put together, joined together to make more than one. So they are a very large chain. That's your amino acid chains, a sugar chain, cellulose that makes up a plant wall, and then amino acids that build proteins. So our carbohydrates are things that we consume for quick energy. Every living thing must need carbohydrates because they're the building block of energies. And we're referring to sugar and starches. Starch are stored in plants, and our sugar we get from our food that we consume during the day's time, and most of those come from pastas and breads, um, rices, any kind of grain um, usually will be considered carbohydrate. Our monomer for carbohydrate is a monosaccharide, and they're formed together to become disaccharides or polysaccharides when you dehydrate a water out of the formula and they combine together. Di, remember, means two, poly means many. So an example of a polysaccharide would be a starch, uh, glycogen, which is how animals store energy. Cellulose is how plants store their energy, and that's what the cell wall of the plant is made out of, is cellulose. Starch looks like a chain link fence, branches and chains. Um, found in Glycogen's found in mammals. Our lipids, remember, are insoluble in water. They're nonpolar. Um, so if you're looking at a liquid that floats on top of water, or a brown substance that forms on top of the water, remember, oil and water doesn't mix. That's why your Italian dressings and salad dressings separate in the refrigerator. You have to shake them up before you use them. The lipids have less oxygen. So examples of lipids are going to be your butters, your oils, peanut oils, um, margarine, but they are used for long-term energy storage, insulation, and protective coating. And so carbohydrates and lipids have in common is that they uh, both are used for energy. Lipids is going to be referred to as being insulation, so that's your key word there. Your proteins are very important. Um, Proteins are referred to as many things in the body. They have lots of jobs. They also contain sulfur, and your monomer for proteins are your amino acids. They're held together, your amino acids are held together by a covalent bond called a peptide bond. Remember, your R group determines the type of amino acid, and proteins are called enzymes. And so enzymes are a type of protein that help carry out your chemical reaction. Um, Proteins build your hair, your teeth, fingernails, your exoskeleton. And the other thing that's very important is they carry oxygen in the blood, and it helps with the immunity. Then we have our nucleic acids. Our nucleic acids are DNA and RNA. They can carry your genetic code. They're very small. Um, their monomer is nucleotides, and your nucleotides are made of a simple sugar, a phosphate group, and a nitrogen base. And those nitrogen bases we'll learn when we study DNA. Remember, this is the diagram we talked about in class. Your phosphate group's always your circle, your sugar in the middle, and then your bases will be on the end. So DNA stands for deoxyribose nucleic acid, and it's a double helix. The strands are formed from your genes and your chromosomes, which give you your traits. Your RNA is ribonucleic acids, and the difference between these two things are the sugars, and that's how they're named, is by their sugars. We also talked about chemical reactions, and this is important to understand how enzymes work. When you have an endothermic reaction, it releases energy, and the energy is released into the environment. Um, your endo is where your energy is released inside, and it's very hard. The example here is heating water. It's very hard to heat water because it has to have an outside source of heat, and for those molecules to heat up and start hitting up against each other. So that's our endothermic reaction. Enzymes, remember, are a catalyst, and a catalyst is something that speeds up a chemical reaction. Remember, enzyme is going to be referred to as an organic compound, whereas a catalyst is going to be referred to as substance. enzymes work to lower the activation rate so that the energy process will speed up. These are your diagrams we talked about in class. Less energy is needed to complete the reaction. So that's what lowering the activation energy means. This is your lock and key model. Remember that the enzyme, the substrate has to fit inside of the enzyme for it to start the reaction. If it doesn't fit, then the reaction is not going to occur. The site that the substance is trying to go into is called the active site, and that's where all chemical reaction is going to occur. Now, enzymes are specific. They have a certain substrate that's going to fit like a lock and key, and that's why we call it a lock and key model for this thing to break down things. Remember, enzymes are used in digestion and to break down food. Any work ending in ASE, we talked about it being a enzyme. So here's our lock is our enzyme, our key is our substrate, and this thing is called a lock and key model. So what causes an enzyme not to work? That's the important part that we need to understand. First of all, when we change its shape, we're causing it to be denatured. And denatured means that we are unfolding the enzyme. So a good example would be taking an egg and boiling the egg. The egg is a protein, but we change its shape from a raw egg to a hard-boiled egg. We denature it when we heat it. So there's a couple of things that will cause enzymes not to work correctly. And the first thing is increasing or decreasing the temperature. So boiling, heating it to where something's overcooked or cooked, and then decreasing it by freezing it. Also changing the pH of the enzyme's environment can also cause the enzyme to denature. Now, when an enzyme is at its optimal pH level, that means that it's at the level that it's going to work the best. Remember, we talked about those and looked at those diagrams where an enzyme is usually between 6 and 8, the optimal level. Those two numbers there, between 6 and 8, is going to work the best. Alright, so that gets us through enzymes and molecules. So now let's go into look at our organelles. And we'll do a quick review of our organelles. So I'll start with the cell theory first, and all cells are composed of one or more cells. They're very organized, and all cells come from pre-existing cells. Our electron microscopes, remember, they go in and we can see more of the organelles very large scale where they're scanned. We can see the nucleus and those things up close, and magnification on them are extremely high. We have two types of cells, our prokaryotic and eukaryotic cells. Our prokaryotic cells lack an internal membrane-bound structures. An example of those is going to be a bacteria. During your lab this week, you looked at a bacteria under a microscope and you saw the DNA in strings and in strands in the cytoplasm. So they don't have a nucleus for the DNA to be housed in. The eukaryotic, you're a eukaryotic cell. So we're talking about animals and plant cells. They have compartments where all of their organelles are structured and close together, and they all have certain places to be within the cytoplasm. Both eukaryotic and prokaryotic cells have a couple of things in common. They have cytoplasm, they both have ribosomes, and they both have membranes. So those are the three things that they have in common. Remember, your nucleus is your